Okay, all right. So hello guys, again, welcome back to the continuation of our microscopic examination of urine pre-recorded lecture. Now, first we'll start with the first sediments. Okay? Before that, again, sa itong two dyes na itong gamiton for automated microscopy, you have phenanthridine and carbocyanine. Kinsa ganitong mustain sa DNA, letter D. Kinsa itong na letter D. Yes, gusto nyo ng mga D, charot. Phenanthridine. And carbocyanine are for mga mitochondria and other structures. Unsay colors of phenanthridine? Orange. And carbocyanine, its color? Green. Okay, wag kalimutan, please. Unsa gani ang volume sa sediment na itong i-examine under the microscope for microscopic exam? 20 UL or about 0.02 ml. Okay, all right. So again, we'll now start with the first constituents under microscopic exam of urine, which are, again, your cells. Now, we have different types of cells. First, you have the blood cells, RBCs, WBCs. Next, you have the epithelial cells, may it be squamous, transitional, or RTEs, renal tubular epithelial. And you have microorganisms in the forms of bacteria or yeast and parasites. And lastly, spermatozoa. Okay, so these are the cells found usually in your urine. Okay, so ready ka na? Handa na ba kayo? Okay, so we'll now start. Okay, so we'll start first with the blood cells. Number one is, of course, your red blood cells. Now, according to Strassinger, red blood cells are the most difficult for the students to identify. Why? Because again, your red blood cells can have different variations in their structures. They can have different kalookalikes, mga kalookalikes, yes naman, showtime na din eh. Kalookalikes with other urinary constituents or sediments. That's why lisod siya i-identify for a beginner or for a student daw. Okay? Alright, so your appearance usually is pale biconcave anucleate discs. So pale siya, biconcave anucleate disc. So lahira kayo siya sa hema ninyo na red blood cells because your hema red blood cells, they are color red, diba? They are stained manggod. But in our RBC sa ihi, usually they're not stained. So medyo lisod yun siya makita niya, gagmay pag yun sila. But generally, their appearance is, of course, biconcave sila, diba? Biconcave, mas like donut, na ay central pallor. Pero di na ka siya makitaan sa urine, diba? Ang central pallor. And anucleate, wala sila nucleus, of course. And faint colorless circles. Now, we have different terminologies. Okay, if faint colorless circles sila, then they are known as shadow cells. Now, what do you mean by shadow cells? These are the red blood cells na nilayas na, or like for a long period of time na murag, ni-disintegrate na siya. So these are your RBCs. They are termed as your shadow cells. Okay, faint or colorless circles. Next, it could be small, rough cells, crinkled in hypertonic urine. Then that's what you call your crinated cells. Now, ngano siyang mo crinkle, di ba? Recall hypertonic, mga hypertonic, hypotonic, di ba? When you say hypertonic, the solution is highly concentrated, right? Okay, and if yung cells na andira, what will happen to the cells? Now, let's say daghan kayong concentration sa gawas than sa sulod, alright? So what the cells do usually, ang buhaton is, mupagawas siya water. Because again, remember, your water is, your cell membrane is semi-permeable to water. So water ang daling makagawas. So, and the solution or the cells really always aim for homeostasis or balance, di ba? So, para ma-equalize ning taas kayong concentration sa gawas kaysa sa sulod, mupagawas siya og water. Okay. Now, what happened? Paggawas og water, muna siyang kuyos or like crenate. That is why na yung mga crinkles, muna siyang crinated cells. Any type of cell po, di ba, under kaning basic, di ba, basic na principle on hypertonic and hypotonic. Now, for the next part, you have lysed or empty cell membranes. In hypotonic urine, you have ghost cells. Now, what happens? Hypotonic. When you say hypotonic, the solution, di ba, or the liquid, or example, the urine, kung asa ang RBCs, hypotonic. Hypotonic meaning decrease iyahang concentration. So therefore, decrease ang concentration sa solution or sa ihi outside. So your RBCs, mas daghan ang mga solutes na naa sa sulod. So what happens para ma-equalize, the water surrounding the solution will go inside, di ba? Para murag ma-dilute ang solutes. Para mo equalize. Because again, they want balance always. So what happens, sige ragpasulod og water, munang buto, mulais. Okay? I hope na gets na ang principles sa hypertonic and hypotonic, ha? Okay? Alright? And when the RBCs lyse because of hypotonic urine, may mo silang ghost cells. Okay? Alright. And if na mga protrusions or fragmented, okay, na mga like different mga characteristics, protrusions ba or fragmented, usually makita na siya, this is what you call your dysmorphic RBCs. Kay murag dili na mao iya mga characteristics ganang fragmented, and usually makita niya siya in glomerular bleeding. So if na ay bleeding sa imuhang glomerulus. All right? Okay. So glomerular bleeding because again, as your cells, diba, from glomerulus mo pa siya sa ihi, daghan pa siyang agian. So pag agi niya sa different areas ato, pwede siyang ma-subject to different stress, different environment, munang fragment siya or na mga protrusions. Okay? So again, dysmorphic RBCs, usually indicative ang dysmorphic RBCs of glomerular bleeding. Okay? And identification error, like asa siya pwedeng masayop o identify, yeast cells, oil droplets, and air bubbles. Because again, they look quite the same. Okay, natay pictures in the next slides for that. But again, asa siya pwedeng masayop, yeast cells, oil droplets, and air bubbles. Muna siyang, according to Strassinger, RBCs are the most difficult to identify or recognize by a student in the CM laboratory. Okay? Normal values about 0 to 2 to 0 to 3 cells per HPF. Okay, we will have a summary na sa reporting at the later part of our presentation. So don't worry. Nakoy mga mnemonics, ana. Okay, all right. Ayan. And clinical significances, of course, renal disease, lower urinary tract disease, mga physiologic causes, and extra-renal disease. Basta usually, if na RBCs makitaan sa urine, most of the time, it could indicate mga bleeding, okay, within your urinary tract. But it can also cause mga physiologic causes lang, like example, mga strenuous exercise, mga stress, or pwede po siya mga toxic reactions that can produce or that can damage the mga tubules, alright, that could result to bleeding, okay? Alright, that's for the of RBCs. Please take note of these, guys. Mga terms. I hope na sabtan lang hypertonic, hypotonic, ha? Okay? A general principle of that, okay? And for some pictures, this is your RBCs. May tagin-ani siya sigitanan in sa kada urine specimen mo. Na yung ba na dili in ana katsada na makarogin siya na RBC. Lisod kayo siya, as in, like ako personally, maglibog pa ko asang RBCs, ana. Kay lisod kayo siya, pod i-identify. Pero usually, ana siya, di ba? Biconcave na ay anucleate mga discs, okay? Alright. And this is an example of microcytic gagmay, and crenated RBC. So as you can see, nang lice na siya, gagmay na kaayo sila. But yeah, for RBCs usually, characteristic yun, makita nimo ang murag na ay buslot sa tunga, which is the central pallor, di ba? Pero yeah, dili siya central pallor diri. Okay, what may stain, all right? But yeah, that's for RBCs. All right? Now, this is a dysmorphic RBCs. As you can see, na yung mga fragments na siya, na yung mga protrusions. Okay? And as you can see, di ba, this is an example of a yeast cell. Ayan. So, medyo same siya sa RBCs. But as you can see, guys, your yeast cells, mga good, are in a way connected to each other. Example, kani siya, yeast, and then ay gamay na protrusion. This is what we call a budding yeast, meaning this is like a yeast na gatubo. This is the mother, and then muni siya iyahang murag daughter cell, okay? So basta makabante ka na murag connected sila to each other, then ay murag gagmay na mga protrusions inana. That could be a yeast, okay? All right, meaning connected sila kay mga budding cells ba nagbud, okay, from the mother, and then magbud sila. So medyo napas like connected na murag neck or yeah, something like that. Okay. Alam mo ko sa term. All right, okay, but nasa mycology ninyo. Okay, all right. And this is an oil droplet. As you can see, di ba, meron siyang RBC, but it's quite big naman. So, than an RBC. So, basin, dili ra mo usahay dili mo malibog, but namay uban na oil droplets na gagmay that could really look like an RBC. Okay? So, how do you differentiate lang? Your oil droplet is quite refractile. Like, if imo siyang move imong slide, murag usahay na ay murag sakto ba ko? Murag na ay light na mupas, na murag mo lihok-lihok ani because it's refractile. Okay? Ang imong oil droplets. And last one is of course air bubble. As you can see, air bubble ka na sa pagbutang sa cover slip and ana. Okay, so again, that's for red blood cells, considered to be the most difficult cells na ma-identify under the microscope for a student daw. Okay. Next, we have neutrophils, of course. Your neutrophils, granular siya with multi-lobed nuclei. Okay. If, again, they're exposed to hypotonic, di ba, mo swell ang cells because magpasulod ra sige og water, kay di ba, hypotonic. So, when that happens, the Brownian movement of their granules, okay, will appear as if they are glittering. Muna siyang, they are called glitter cells. So glitter cells are WBCs lang in hypotonic urine. And the granules appear to be glittering because of their Brownian movement. Okay. And we have what we call your stain, Sternheimer-Malbin. Now, in your Schlesinger also, there is a table for all the stains na ginagamit. Usually, kaning staining sa microscopic exam, we do it for confirmation or for further identification if dili kayo ta sure. Okay? So, wala lang siya na-appeal din. But that will be in your lecture. But we'll just maybe mga stains na i-introduce din, depending on the cell. But for neutrophils, we use what we call Sternheimer-Malbin. Usually, general stain siya for microscopic exam. Not only neutrophils yung ma-stain, but other constituents. Pwedeng casts, pwedeng mga cells, epithelial cells, but please take note unsa iyang composition, crystal violet and safranin O. Muna akong pag-remember sa Sternheimer-Malbin, gram stain siya na composition. Diba? Kaya atong gram stain, diba, crystal violet and safranin ang iyang primary stains. Muna, so unsay composition sa Sternheimer-Malbin, you have crystal violet and safranin O. Now, sa board exam, ni gawas daw, unsay question, what type or what is a Cova stain? Ayan. Cova stain, guys, if makabante mo sa Schlesinger sa mga pictures, Cova stain is a type of Sternheimer-Malbin. Okay? This is just a commercial product or commercial name. Pero unsay hang stain, it's a Sternheimer-Malbin. Okay? Please take note. Again, Sternheimer-Malbin is not limited to staining neutrophils only. Pwede siya makastain of other mga cells, mga casts. Okay? Alright. And again, what is a Cova stain? Cova stain is a Sternheimer-Malbin stain. Cova stain lang is a commercial brand. Okay? Or product. Alright? Identification error, pwede siyang mahimong RTE cells. But please take note, your RTE cells are not granular and the nucleus ani is eccentric, meaning wala siya sa center, na siya off-center, gamay. Ang RTE, will go there later pag abot sa epithelial cells. And normal values again, 0 to 5 or 0 to 8 cells per HPF. And clinical significance is, of course, infection or inflammation of the genitourinary tract. Now, if you have many WBCs, increased WBCs in urine, that is what we call as pyuria. Okay? Pyuria. Increased WBCs in urine. So if nakay pyuria, increased WBCs in urine. Okay. Alright. So here example of neutrophils. As you can see, nasa yung nucleus, di ba? Nucleus, and then nasa yung mga granules sa kilid. Alright. And yeah, those are your WBCs and glitter cells. As you can see, na ay sidlak. I'm not sure if you're all makanotice mo, pero nice sidlak around the cytoplasm. Yeah, so that's your glitter cells. That's for neutrophils. Okay. All right, now we go to eosinophils. Now, your eosinophils, they are not normally found in your ihi, okay? And can only be seen lang na siya or can be stained if you want to visualize it using the Hansel stain. So, if na question, what is the primary stain of choice for eosinophils in urine? Hansel stain. Unsay composition sa Hansel stain? Methylene blue and eosin Y. Ayan. Unsay kani composition sa Sternheimer-Malbin? Gram stain. Ayaw answer gram stain ha, pero unsa to? Safranin O and crystal violet. For Hansel stain, it's methylene blue and eosin Y. Again, para asan ni siya? For eosinophils. Ayan. As you can see. Na ay mga eosinophilic granules. Now, again, these eosinophils are not commonly seen in the urine. And if naman gani gagmay ra gyud sila na amounts. But when they are in greater amounts, if it's greater than 1% of the WBCs, in a 100-500 urinary WBC diff count. So, we perform a WBC diff count if makakita tagdaghang eosinophils. Because again, it's quite significant if naakay eosinophils sa urine. Because again, it's not commonly seen. Alright? Or gagmay ra ang naa dito. And usually what we see is, we see it during drug-induced interstitial nephritis. Now, basta gani guys, do not, ayaw rag ka worry or like, wag na mag-isip. Basta gani, ang question kay, naay presence of eosinophils sa urine, press the buzzer, the first disease na pwede yung musulod sa imong mind is drug-induced interstitial nephritis. Now, what do you mean by drug-induced interstitial nephritis? As far as I can remember, medyo basin sayop ko ani. Basin, di na ako ma-explain tarong yun. But when you say drug-induced, it's because of a drug na imong gitake and then ni-inflame imuhang murag tubules, I think, sa imuhang kidneys. Alright? In a nutshell, I think mo na siya description. Okay? Ang drug-induced interstitial nephritis. So, murag in a way, murag siya allergic reaction to the drug sa na imong giinom. And dito sa kidneys or sa tubules ni manifest. Okay? Munang na-present sa eosinophils. And another disease or another significance niya is renal transplant rejection. Now, in renal transplant rejection, if na-present sa eosinophils, it means gi-reject ang example ka nang nagpa-kidney transplant. So, na-rejection, meaning wala gidawat sa body. So, therefore, the organ is not functioning well. It could die, okay? Or it could, it could die. It could not function well. So, munang, kailangan nato siyang i-quantitate, okay? Alright, so muna siya ang eosinophils. Basta gani greater than 1% of WBCs in a 100 to 500 urinary WBC diff count. So, if greater than, example, 250 imuhang diff count, so greater than 1% ana, so sa may 1% sa 250, mga 2.5? Oh my gosh, Mark. Basta, okay, mahina ako sa math, pero yeah, greater than 1% of the number of diff count mo na WBCs, significant na ang imong eosinophils. If you see more than the 1%, atong diff count nimo. Okay? Alright. So that's for eosinophils. Again, unsa na stain? Hansel stain. Unsa ganito na disease or condition, first thing mo sulod sa mind, if madunggan gani ang eosinophils, could be, again, drug-induced interstitial nephritis. Alright. Okay, so that's for the blood cells. Medyo gamay lang, di ba? From our red blood cells, white blood cells, and yeah, red blood cells and white blood cells. Now, we go now to your epithelial cells. We start first, of course, with your squamous epithelial cells. It's the largest cell in your urine sediment. And it's really prominent po, actually prominent nucleus and abundant po siya. Because again, your urine passes through a lot of tissues or like passes through urethra, which contains a lot of squamous epithelial cells, right? So, muna siyang its importance na mag-midstream clean catch ta para ma-minimize ang external contamination, right? Okay, so again, squamous epithelial cells, kabantay naman mo sa itsura sa inyong epithelial cell, sa inyong histopath, right? Okay. Identification error niya, the folded, pwede mo siya ma-fold, they may resemble as casts. But please take note that your casts do not have nuclei. Okay, so if nuclei na siya niya, medyo mura siya cast, then that could be a squamous epithelial cell. Now, usually magod since dako kayo siya, dax siya. If gasugod pa lang kaglantaw, to determine na you are in the right field of view sa microscope, if makakita ka og squamous epithelial cell, then that is already indicative na you're in the right plane of view sa microscope. Okay? So, unsa ganyan yung pangitaon para ma-determine if you're in the right plane of view or like right plane sa microscope, a squamous epithelial cell. And if makakita ka, then you're in the correct plane of view na sa microscope. Okay? Alright. Clinical significance niya, of course, no pathological significance usually ang squamous. But we have these particular cells known as your clue cells. Wow, yes. Clue cells, usually makita niya siya in bacterial vaginosis. Okay? Caused by what organism? Gardnerella vaginalis. Okay. Muna siya causative agent sa bacterial vaginosis. Ang Gardnerella vaginalis. Okay? Alright, now what happens is, in clue cells, ay muhang epithelial cells, they are laden, heavily laden. So, example, epithelial cell ni siya. May murag sunny side up. Sorry, guys. Dagdagan kayong bacteria, gyud, na maabot na gyud sa gawas, na palibot niya. That's a clue cell, okay? And indicative na siya o Gardnerella vaginalis infection or bacterial vaginosis. But most of the time, non-pathologic ra ang squamous epithelial cell. Unless na lang makakita kag clue cells. Again, which is indicative of Gardnerella vaginalis infection, bacterial vaginosis. Okay, so here's an example of under LPO, di ba, daho kayo siya, makitaan gid siya dayon. Muna siyang ginagamit siya as, murag in a way, our reference, point of reference. If chakto gid ta, we're on the right track, baby, I was born this way, joke. We're on the right plane of view or plane of field sa microscope. And again, under HPO, nag-clump. So, dako jud kayo siya na cell, center ang nuclei. Okay? Alright. Next, we have your transitional epithelial cells known as your urothelial. Diba? Back in your anatomy, these are mga epithelial cells na pwedeng mukuyos if walay fluid, diba? If wala sila gisudlan, o walay musulod nila, and then mu expand na po sila. So they're capable of expansion and kanang retention ba na? Right? Diba? Usually makita siya sa bladder, ureters, and mga upper portion of urethra. They can be seen as in singly, like usara gyud, or in clumps. Please take note, clumps, syncytia. Okay? Alright, syncytia. Again, appearance could be spherical, polyhedral, or caudate. Caudate meaning na ay tail-like appendage. And again, ilahang nucleus is centrally located. Identification error, ang spherical forms niya may resemble as an RTE, okay, or renal tubular epithelial cell. Okay. And clinical significance niya, increased numbers lang with abnormal morphology if kanang naay mga in malignancy or viral infection. And under invasive procedures, example ka ng catheterization, kaya di ba, mapasulod man sa urethra, padulong sa bladder, so that could lead to the shedding of many transitional epithelial cells. So if it could lead to, again, invasive procedures, example, catheterization, and na-increase sa imuhang transitional epithelial cells. But usually, again, they are seen from mga pelvis, kana nasa bladder, usually sa lower urinary tract, di ba? Okay? Yeah. So these are again transitional epithelial cells. And the shape depending kung asa siya gikan. Okay? Alright. So here's an example of polyhedral, di ba? Usually magkumpol-kumpol sila, guys. Pero actually same na po siya as an epithelial cell or squamous epithelial cell, di ba? So yeah, nagkumpol-kumpol sila and pwede po niya siyang stained na spherical transitional epithelial cell. Medyo smooth ng ilahang border, okay? Right? And yeah. So basically, that's a transitional epithelial cell. And this is what we call a caudate, di ba? Na siya yung tail-like appearance. Pero that's an epithelial cell. That's transitional epithelial cell. And next one is kanisha, syncytia, meaning nagkumpol-kumpol. Asa siya gikan? In a catheterized specimen. Because again, invasive procedure. So pagsulod sa catheter could be na daghang mga shedding of transitional epithelial cells because, again, invasive ang procedure. Okay, so if na-increased in transitional epithelial cells, could be indicative of invasive procedure. Or unsay makit-an na daghang cells in an invasive procedure such as catheterization, transitional epithelial cells. Okay? Alright.